Hello class, today we are going to cover Punnett squares. So the genetics portion of Punnett squares. Let's go ahead and begin. So let's consider what a gene is. Now this is a cell, this is the nucleus, cell, cell, this part is the nucleus, this is a tightly packed chromosome, this is an unpacked chromosome, and this guy is one gene. Now you can see from our DNA section that here is our deoxyribose sugar base mixed with our phosphate base that form the backbone of the double helix. Okay, and these multicolored things again are nitrogenous base pairs, and they are complementary, which means A always goes with T, G always goes with C. And you can see that a gene is a segment of those nitrogenous base pairs. So based off of which pairs are ordered in which order, that will determine the gene that is expressed, okay? And then the gene gets expressed and it makes a protein. So that is the gene. Genes are located inside of the chromosomes. Chromosomes come in pairs, and there are thousands of genes on each individual chromosome. This portion of the lecture will serve mainly as a review. You should know and be very familiar with everything I just said and the next slide that I am going to say. So in humans, a cell's nucleus contains 46 individual chromosomes, or 23 pairs of chromosomes. Half of the chromosomes will come from one parent, and the other half will come from the other. So this picture is a karyotype. For those of you who did this and missed this on the quiz, which many of you, this was the most common thing missed, which is why I put it on this video to reiterate it. The karyotype is this picture representation of all the chromosomes in a species. For this case, this is showing 23 pairs or 46 individual chromosomes, and it's a male because we have a Y chromosome. If we did not have a Y chromosome, it would be a female. If we have a Y, it's a male. Okay? Hopefully that is concrete now. This is a detailed structure of your chromosome. As we talked about in our mitosis discussion, this central area is chromatin, and then during metaphase, the spindle fiber goes from the centriole and attaches to a kinetochore. So this would be your kinetochore. And the kinetochore is a sticky-ish protein substance that attaches itself to the centromere. Okay? So let's get into our genetics recap. Again, we have talked about this in class. So if this is new to you, you need to come see me if you are confused. An allele is a discrete version of the same gene. So is it purple or is it white? Okay, one allele is expressed by one letter. So big P or little p. Okay, that is an allele. The genotype is the genes of an organism for one specific trait, and it is typically written with letters. So 2 capital P's is homozygous or homogeneous dominant for purple. Okay? Phenotype is the physical appearance of that genotype. So big P is dominant, homozygous, I'm going to see purple. Big P, little p is heterozygous because, again, remember, hetero is different capital letters, different, hetero, okay? Big P, little p, it's still got a big P, so as long as you see a capital letter in your genotype, you are getting the dominant trait. So big P, little p, you are purple. And then again, you have another opportunity for a capital P and a little p. And then for your 2 lowercase p's, that is when you are recessive. And in this example, the recessive trait is white. Okay? So phenotype is the actual physical description. Genotype is the individual gene makeup, and it is shorthanded by these letters. So again, to recap, this is for a human trait for your earlobes. We talked about this in class 2. Dominant trait refers to the genetic feature that hides the recessive trait in the phenotype of the individual. So homozygous, same. Homo means same. So same capital letters, big E, is dominant, which means it shows detached earlobes. Heterozygous, capital E, little e. Hetero, different capitalizations, will still give me a detached earlobe because, again, anytime you see a capital letter in the genotype, you get the dominant phenotype. And finally, you have recessive alleles that are homozygous, little e, gives an attached earlobe. Okay? So that is your recessive trait. And finally, we get to put some of this stuff into practice. So we know that we use 2 letters to represent the genotype. A capital letter represents dominant. The lowercase letter represents the recessive form of the gene or allele. Okay? So for example, if we are homozygous dominant, we have 2 capital P's. If we are homozygous recessive, we get 2 little p's. And this finally leads us into today's topic of Punnett squares. And a Punnett square is the standard way of working out what the possible offspring of 2 parents will be. It is a helpful tool to show allelic combinations and predict offspring ratios. So what you need to know for your test is that a Punnett square is the standard way of working out the possible offspring of 2 parents. Okay? So in this example, we have big P little p crossed with big P little p. And we'll show it more in detail in just a minute. You begin constructing a Punnett square by forming a grid structure with 4 boxes in it, as you can see on your screen. Okay? The next step, you want to put the genotype of one parent across the top and the other along the left side. For this example, let's consider a genotype of capital B with lowercase b. So I have a capital B here and a capital B here. And then on the left-hand side, I have a lowercase b and the lowercase b. So the first parent, each allele of this genotype gets broken up into its own column. The lowercase letters, or whatever the genotype is, of the other parent goes along the left side. So this B goes here, this B goes here, crosses with this B, and this B. Hopefully that makes sense. Okay, let me erase some of these. Okay. So now, to fill in the boxes, you want to copy the column and row head letters down and across. So what does that mean in English? Does that make sense? Okay. What I did here is I took this parent capital B and I put it in each of the boxes that were underneath it. So I took it and I put it in this box and one in this box. Because again, we're just trying to figure out probabilities. For this capital B, I also put it in this box and one in this box. So it represents here, 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 and here. Then for my other parent, I do the same thing, but now I do it across. So this B gets filled in and that B gets filled in. Then from here, this B and this B. Capital letters go first. Lowercase letters go second. Okay? Let me show you that animation again, so see if that sinks in some more. Okay. So, capital B's go down, fill in the available spaces, then the other parent also gets filled in. Okay? Capital B's go down. Now these could be lowercase b's too. It doesn't have to be. Just for this example, we're dealing with capital B's. So capital B's go down and fill in the open spaces, and then these B's go over and fill in those spaces. Okay? And that is how you complete a Punnett square. So now that we have learned the basics of genetics, let's walk through some examples using our Punnett squares. So let's say we have a dominant white allele and a recessive violet allele. So the parents in this cross are going to be heterozygous. So each parent is capital W, lowercase w, because heterozygous means different capital letters. So in our Punnett square, you can see we have a capital W and a lowercase w, a capital W and a lowercase w. So our capital W gets filled in here and here, and our lowercase w gets filled in here and here. This capital W then gets filled in here and here. Now, why did this capital letter get over on the left side when this parent was this? Okay, remember, the capital letters go first. So if you have a parent with a lowercase letter up here, you want to go ahead and fill it in on the right-hand side. Okay? Now, this is a lowercase w, and it gets filled in here and here. So again, if this is a lowercase w up here, fill it in on the right-hand side. If again you have another lowercase w, you can then fill it in on the left side, because we usually write the capital letter first. Okay? So this is how you complete a Punnett square with a heterozygous trait. Let's try another one. So, okay, hold on. Make sure I can tell your capital letters from your lowercase letters, which you can see this is a little different. There's a little space here, but not a whole lot. So if I was going to handwrite this out, I maybe would make the W just even smaller than that, somewhere around there. Okay? So what percentage of the offspring will have violet flowers? Remember, violet is the recessive trait, and to see the recessive trait, we must have both lowercase, the offspring must have both lowercase w's. So out of these boxes, there are 4 chances, and only one of them, this one, has 2 lowercase w's in it. So we have 1 out of 4 possibilities will be the recessive violet trait. And if you remember math, 1 divided by 4 is 25%. Okay? So 1 out of the 4 squares will be heterozygous and will show the violet phenotype. The next one. Red hair is dominant over blonde hair. Make a cross between a heterozygous redhead and a blonde. So heterozygous redhead means I've got big R and little r, and a blonde means that I'm recessive, so I have little r, little r. So again, I fill in my big R by taking it and filling it down. Take my little r and fill it in on the right-hand side, because in case there's a parent with a big R over here, there is not. So then I can fill in my little r over here, little r over here, and then this r goes here and here. Okay? So then what percentage of the offspring will have red hair? Now remember, red hair is dominant, so we need to find any genotype of the possible offspring that has a capital R in it. So here's 1, here's 2, and that gives me 50%, because there's only 4 opportunities. 2 divided by 4 is 50%. Hopefully this is getting there, okay? We're going to go through one more, I believe. Let me make sure. I think we're going through one more example for you, and then we will call it, or 2 more examples. Okay. So let's try a couple more. In pea plants, tall pea plants are dominant, short pea plants are recessive. So construct the Punnett square for a heterozygous tall pea plant and a short pea plant. See if you can do this one on your own. Go ahead and pause the video right now and see if you can complete this on your own, and then restart the video once you think you're finished. Okay, hopefully that means you have paused the video and you didn't just watch through and go through the awkward silence with me. Let's see what the answer is. Here we are. So we have a heterozygous tall pea plant, so big T, little t, and a short pea plant. So short pea plants are recessive, so you mark those little t, and then you go through and you mark, here's your big T, you put those down, little t's, you put those down, little t's you put across, little t's you put across. So what are the percentage of the phenotypes? Well, I've got one big T, one little t, one little t, one little t, another big T, little t, and another little t, little t. So I'm going to have two big T, little t's, and two, those are our genotypes. Anytime I see a capital letter, I'm getting the dominant, so that means I've got tall. And these are both short or recessive alleles, so I'm going to have short. So I have two tall plants and two short plants. So 50% tall, let me get rid of mine, and 50% short. Hopefully that makes sense. Okay? And last time, make a cross between a homozygous rat with black eyes and a rat with red eyes. So black eyes are dominant, red eyes are recessive. So we have homozygous black eyes and a homozygous red eye. Now, how do I know it's homozygous red eyes? Because red eyes are recessive, okay? They're recessive, so they have to, for recessive phenotypes, they have to be homozygous. So go ahead again, pause the video, see what happens, try your best, cross this homozygous black eye with a homozygous red eye, and then we'll see if you got it right or not. Pause now. No, seriously, pause now. Don't just sit there and watch it and wait. Okay, I'm assuming now that you listened to me, hopefully, and pause the video. So let's see what we got. We have a homozygous rat with black eyes, so that means capital R, capital R. And I've got a recessive because I have one with red eyes, okay? So when I cross them, I get all heterozygous genotypes, which means I'm always going to see black eyes because black eyes are dominant, okay? So what is the possibility of a red eye offspring? That is 0% because none of these are homozygous recessive. Okay, and now hopefully that was very educational and you got it and you're not completely lost. You have several more problems to do on your own attached to our homework. If you have any questions, please feel free to email me or ask me when we are in class. Thanks very much for watching, and I will see you guys soon.